Hello and welcome to our week 7 virtual lab for general botany. I'm Stephanie and we're going to be talking about applying hypothetical deductive reasoning. In other words, doing science. We're going to talk about how to do that today. You've already learned a little bit about the scientific method and what hypothetical deductive reasoning actually means, but we're going to expand on that quite a bit today. And I want to start off by walking you through this thought process that I recently had with my dog Holmes. We're actually going to kind of pick up from the end of last week's virtual lab where you saw he indeed likes to chew on plants that have undergone secondary growth. And I had always noticed he seems to like some sticks more than others. There are various sticks around in the yard and where we go play, and he has various preferences for them. And I have this hypothesis that he prefers smaller sticks. Okay. Well, I've actually just started doing science here. I made an observation, I had a question, I listed variables, and then step 2 was I made a hypothesis. And I was wondering, is Holmes's preference dependent on the attributes of a stick? And so the next step here would be to go ahead and start designing sort of a way to test this hypothesis. So can I just kind of look at him in my yard and see if he chews on small sticks? Will that will that potentially test my hypothesis? Is he chewing on small sticks? No, that's not good enough. There's no comparison there. Maybe he's just chewing small sticks because that's all that's available and that's the only thing that's woody. No, you have to have something to compare you have to have something to compare your variable to. So then can I just give him one small stick and one large stick and see which one he picks? No, that is also not good enough to fully answer this question of what Holmes's favorite type of stick is. If we just give him one of each, well, maybe he's just choosing the small one here because there was a little bit of honey residue on it. Or maybe this large one had some strange smell to it that he did not like. So just because he chose this one doesn't mean that he always will choose a small stick. That would be an inductive leap. Okay, so then how about we go to the beach where there's lots and lots of big sticks. And we see how he chews on sticks there. And then we bring him to my yard where there's lots of small sticks and see kind of how he chews on sticks here. No, well, that also will not work because those are two completely different environments. And maybe he'll chew on sticks differently at the beach because he's trying to show off around all the other dogs, compared to in my yard where he might be more comfortable chewing on whatever he prefers. So no, they would have to be everything that we need to compare regarding Holmes's preference of stick needs to be in the exact same environment. We have to have many replicates. And we have to have nice comparable groups. So, in other words, when we produce a testable hypothesis, we need to calm down, we need to sit down and design a controlled experiment. That is going to be most of the point of today's talk. So the rest of the steps here, just to brief through them relatively quickly, where the next step is that, okay, I'm gonna need to collect some sticks, I'm gonna need to make some free time, because I just designed this experiment. I'm going to set up the sticks based on this controlled experiment that I've designed, and then I'm going to set them loose. I'm going to record whatever the variables are that I'm recording, which might be his time spent chewing large sticks versus small sticks over some extended period of time with multiple replicates. And then I find out, did he chew small sticks a lot and not large ones, or did he choose large ones and not small ones? Whatever it is, I need to find out. And then the last step would be to conclude. In this case, what I'm pretty sure would happen is we would conclude that Holmes does not prefer large sticks. Notice, I'm not saying that he prefers small sticks. I'm not saying, he loves small sticks, he's always going to pick a small stick. No, all I'm saying is, compared to small sticks, Holmes will not choose a large one. Okay. Over small ones. So that was actually doing science. Even though I breezed through most of those steps, if I had run everything that I just explained soundly, then I could have been doing science. So what we're going to be talking about for the rest of this video is how to apply hypothetical deductive reasoning to one explanatory model. Now, we talked about in the scientific method video, the lecture video from this week, that hypothetical deductive reasoning is sort of one way to use the scientific method. And we talked about how when you have an observation, there could potentially be multiple models that are explaining your hypothesis or your observation. Either one. And what we're going to be focusing on is just one single explanatory model and how to falsify a hypothesis. So what I've done here is I've made these steps. I've sort of manipulated other versions of procedures on how to falsify a hypothesis. I've manipulated them to sort of perfectly fit with the purposes of our week's virtual lab goals and this week's lab assignment goals. So I've sort of expanded on some ideas. I've sort of contracted other ideas together. You'll see. Okay, step 1. You make a list of observations, questions, and variables. So here's an example. Maybe some other version of the steps of this process would have had those as 3 separate steps. I've put them all together because they kind of go together. You have an observation. It causes you to think of a question, and then you have to sort of sort out what are the variables of your question. Once you've done that, you form your hypothesis and your null hypothesis. After that, you need to design an experiment, but this is arguably the most important part, because the rest of these steps will be dependent on, your conclusion will be dependent on the soundness of this experiment. So your experiment has to have comparable groups, it has to control for external variables, use replicates, randomization. These are going to be the main focus of the rest of this video. Step 4 would be to make a list of all the materials that are needed, and then you actually set it up and run the experiment. You take the measurements, you get results, you analyze your results, and reject or retain the null hypothesis based on your analysis. And then you make a conclusion. Spread the word. All right, the truth is that this lab, and spoiler alert, your lab assignment, will be focusing on step 3. And I'm going to do something absolutely crazy for steps 5, 6, 7, and 8. And I'm just going to completely make them up. We're just going to make them up on the spot. They're going to be fake results, fake conclusions, but we're going to pretend like they're real. This is all hypothetical. So what are the basics of what we need for an experimental design? We need a testable hypothesis. We need an independent variable that we can manipulate or that has been naturally manipulated for us. And we also need one dependent variable that we can measure. Okay. So we're going to walk through one example of the whole process of falsifying a hypothesis now. The focus will be on step 3, the experimental design process. Okay, so I see this trend all over the place, not just in one place, not just from this one picture. Stems and leaves that are growing in shaded areas are less robust than their counterparts that are growing in sunny areas or in full sunlight. I've just noticed this across many different species of plants and in all different types of places, honestly. And so this picture sort of represents that. We've got these 2 plants. They're whatever plant they are, whatever species they are, they're the same thing, same species. And this one, it's growing under the shade. That's sort of what this gray represents. This is under a tree. That's a tree. And notice it's smaller. This one over here, it's growing in the full sun. Notice it's larger. This is my observation. Well, it made me wonder, what's going on underground? Are the roots also apparently constricted or restrained by sunlight intensity, like the stems and the leaves are? I wonder. So my question, which is still part of step 1 here, is root growth affected by sunlight intensity? There are always at least 2 variables associated with any solid experiment. You've got an independent variable. This is what you would be manipulating or what is causing an effect. And so, I'm wondering, is the shade sort of causing an effect here? And then the response, the other variable in my situation here, is the response variable, which is also known as the dependent variable. And this is, what would I be measuring? Or what is actually different because of my independent variable? I've got 2 variables here. I've got root growth, that's what I'm wondering about, and sunlight intensity. I'm wondering, is root growth dependent on sunlight intensity? Or I could be saying this is root growth dependent on shade intensity. And so root growth, I'm asking if it's dependent on something, and so it is the response variable, while sunlight is the independent variable here. Okay, so the next step is to turn this question into a testable hypothesis. A hypothesis, it must be a full sentence, it must be testable, and then we'll create a null hypothesis. So my hypothesis here, based on my observation and my question, is the roots of plants that are growing in low intensity light, shade, will have root systems that weigh less than roots of plants that are growing in high intensity light, which is full sun. Okay, so we need to have a null hypothesis to make a counter argument, essentially. Just to make sure that whenever this whole process is over, we're not going to end up making any inductive leaps. We need to keep it deductive by having a null hypothesis, which we can falsify. So the null hypothesis here is, if shade does not reduce root growth, then the roots of shaded plants will be equal to or heavier than those growing in full sun. Those roots growing in full sun. Okay. Now we're going to design an experiment. This is the main focus of today's lab. So an effective experiment will have many things. Here are 3 of those things it will have. It will be able to precisely manipulate the independent variable. It will be able to precisely define the groups of the independent variable. So treatment groups and or control groups. And it'll have comparable groups. This is very important. So let's expand on those. So I'm wondering about shade versus full light. And this is the really kind of exciting part of science where we get to be creative. We have a question, but imagine there are countless ways that one could go about answering this question. This is just what I would do. Maybe I would I would build an apparatus that can cover certain plants, and that would represent shade, and the other ones I would just sort of leave unaltered, and they would represent the full sun. Okay. So, just a reminder, the independent variable is light intensity. The dependent variable, I'm choosing root weight, because I had originally kind of wondered about the robustness, and I thought weight was a good representative of that. Could I have chosen length, though, as the dependent variable? Sure, I could have. Maybe it would sort of depend on how these plants grow. I would want to look into that before I make a decision on what exactly it is that I'm going to be measuring. I've just chosen weight here, and I'm good with that. I'm going to plan on having 2 groups. I'm going to have the shaded group. This will be my treatment group, where my potted beet seedlings will be covered by this netted apparatus thing that's only going to let 25% of the sunlight through. It's going to block 75% of the sunlight. So again, this is where I'm just being creative here. I could have chosen 50% of the sunlight that it'll block. Or I could have, let 75% of the sunlight through and it only blocks 25%. Also, I could have had more than 2 groups here. Imagine a scenario where I have 10 different groups, and in some group, or in one group, there's no covering. In the next group, there's 10% of the sunlight that is blocked. The next group, there's 20% of the sunlight blocked, and then 30% of the sunlight blocked, all the way up to 90% of the sunlight blocked or something. Could have done that. For simplicity's sake, I decided to go with 2 groups. Cool. Right. So, other things that an effective experiment will do. We already talked about sort of number 1 here. Precisely manipulating the independent variable and defining those groups. And these groups must be comparable. And so by comparable, what we need to make sure that we're doing is controlling for any outside variables. Number 2. We're also going to need to use replicates. Just sort of bringing it back to earlier when I was going to just present Holmes with 1 large and 1 small stick. He could have just chosen one because it had some smell to it, not because of its size. You need to make sure that you're controlling for all external variables by using replicates, lots of them, as many as possible. And then we need to precisely measure the dependent variable. So let's expand on these a little bit. Speaking of that number 2 from the previous slide, controlling for external variables, what we need to do is use randomization wherever is needed throughout this experimental process in order to prevent unsound results. So in this experiment, I'm going to use a bunch of plants in theory because I'm going to use a bunch of replicates. And so in order to do that, I'm going to germinate some bead seedlings. That'll be my very first step, maybe, is I'm going to kind of start the germination process of some bead seedlings like you see in this picture. What I should not do is put these bead seedlings in order from largest to smallest, and then put all of the largest ones into the experimental treatment group, and then all of the smallest ones into the control group. That I should not do. No, instead, I should be randomly choosing which seedling ends up in which group, right? Because if all of the largest ones end up in one group and the smallest ones end up in the other group, when I'm supposed to be testing for shade versus full sunlight, maybe the results that we would get after doing the shade experiment would actually have been because of the size of the starting seedling. We need to control for all external factors and have everything be as identical as possible between replicates, except for the one thing that we're manipulating. So here, except for the fact that some of them will be covered by a shaded covering. Okay. Step 3, factors that will be controlled. So some other things that I need to control for here. Since I'm going to be weighing roots, and since roots will have different levels of, different amounts of dirt attached to them, we need to avoid the varying weight of dirt stuck to different roots. And so I'm just going to grow them directly in water to have clean roots. So they'll all be growing in a hydroponic system, the same hydroponics system, so that the water, the temperature, the ambient air temperature, everything will be the same, including the potential exposure of insects, the wind, everything will be the same. All seedlings will be of the same bean species and the same cohort, meaning I sort of started them all out at the exact same time, and were assigned randomly to groups, treatment versus control. Roots must have dried for the same amount of time. Every single root would need to be completely dry. Maybe you've let it dry for 24 hours or something. Again, you've kind of got to do your background research and get creative here. Or get creative here. Okay. So here's maybe something I would do. Maybe this is what it would look like. I would have, this is sort of my whole thought process of my experimental design still, right? We're still actually just in step 3 of designing an experiment. And here, since it's a hydroponic system, what I've done is I've put these plants, these pots in holes on a styrofoam. That's what this gray represents, styrofoam. So they're all floating, and they're all receiving the exact same nutrients and water. And, hopefully I would have plants sort of equidistant away from this water and air pump, or at least I'll have an equal number of treatment and control groups by this pump. So you've got to really account for all of the things that could affect results. How will I let them grow? Will this be indoor, outdoor? If it's indoor, what type of lights will I use? What exactly will this shade netting be made of? These are all questions that I would have to get creative to answer. And, how many replicates do I want? Is 4 enough? No, it's not. Let's do 40. Yeah. 40 sounds much better. Just a good rule, a generalized rule is in science we tend to use n equals 30, a sample size of 30 for the minimum kind of good sample size. Although if you can ever do more replicates, that is always preferred. So I chose 40, 20 of each category here. And so here, these gray squared ones represent my shaded treatment group, and then these brown circles represent my control, full sunlight group. Moving on. All right, now I'm going to be breezing through the rest of this because this is where it gets really hypothetical. Step 4, actually, this is less hypothetical, I guess. You could still really make a list of materials. So everything that I just mentioned using, and I didn't even mention certain things. Everything that you could possibly imagine needing for setting up this experiment. Make a list of that. And then step 5, set it all up and run the experiment. Very exciting. We're not doing this in real life. Imagine this being done hypothetically. Hence the little thought bubble. Okay, step 6 is to take the measurements. Record the data. Excuse me. So here's one of these plants that has either, I can't tell from this picture if it's from the treatment or the control group, but it would of course be labeled. All of these samples would be labeled 1 through 40 and either shade, S for shade, or C for control. And then I would just sort of take a razor and slice the whole shoot system off and measure the dried roots for each and every one of those 40 specimens, labeling both the weight and which treatment group it was in. Was it in the shade or was it in the control group? Step 7 would be to analyze the results and either reject or retain the null hypothesis. Remember, by accepting the alternative hypothesis as truth, we could accidentally be making some inductive leap in the case where there actually might be some other explanation for our conclusion. But instead, by just rejecting the null, we've falsified one model that could explain our observation. So we've used deductive reasoning to gain support for our alternative hypothesis when we reject the null. So here, remember, the null was if shade does not reduce root growth, then roots of shaded plants will be equal to or heavier than those growing in high intensity light. So you need to analyze your results. Again, just a picture for what step 7 might sort of look like. You've got your data sheet, and then you make some sort of figure that shows your results. This I've just whipped up in a second, just for the purposes of our lab this week. I've got a title, and I've got the X and the Y axes labeled, and yes, those X and Y axes do jive with what we call the X and Y variables, the dependent and independent variables. So remember, we were wondering if the root weight is dependent on the independent variable, which was light intensity, X. And then we make a conclusion and we report it. This is a very important and exciting part of science, sharing it with the world, communication. And since this was all hypothetical, that is it. Now I have a little bit of a spoiler alert for you. Spoiler alert. You will be designing an experiment with your group for the lab assignment for this week. Here, during this whole video, I have kind of found out if root weight is affected by something, namely sunlight or shade. You will be finding out if the rate of transpiration is affected by something. And so you've been learning about transpiration, you've been learning about experimental design, and you're going to put those together. So here, in my example, I manipulated the environment. I created shade. For you, you're either going to manipulate the environment, like maybe create shade, or create wind or heat or something. Or you can manipulate the plant itself. So maybe you want to know if the leaf shape has an effect on the rate of transpiration, or adding trichomes in different places. We're going to give you this whole extensive hypothetical toolkit, which will be sort of longer than the one you see here. And I just want to really encourage you to be creative. So the assignment is posted, and you can read about that. Have fun with it, and let us know when you have questions. Bye.